All right. Thanks so much for the invitation. As just mentioned, I'm going to talk about a few different projects today, but the overall theme of what I'm going to be talking about is how noise impacts neural computation. And what I hope to convince you of is that noise has really subtle and interesting consequences for neural computation. And from my perspective, at least, it's something that I think is still pretty poorly understood outside of really simple toy cases where we can get a good handle on it. So I'm going to talk about a couple new computational tools and perspectives my group has been developing, which I hope is going to help us make more progress on this problem. So this is a workshop on neuroscience, psychology, and AI. So there's many ways to motivate why noise would be important to all these communities, but I'm going to start by talking about some work, really centuries-old work in many cases from the field of psychology. So specifically within psychology, within the field of psychophysics, there's lots and lots of experiments that go something a little bit like this. You show a sensory stimulus to a human participant, and then you ask them to tell you something about that stimulus. Like, is this tilted to the left or tilted to the right? And if I flash a stimulus like this really quickly on the screen and I make it close to vertical, I can make this arbitrarily difficult. And what you see is that humans are pretty good at tasks like this, but their performance degrades as you make the task more and more complicated. So how do we build a model of that system? Well, the more or less accepted principle or model, working model within psychology for a long time has been that this external sensory input is somehow mapped to an internal representation that lives somewhere in the brain. And the fidelity or noise level within that internal representation sets a fundamental limit on any sort of downstream behavior that needs to use that sensory information. Okay, and of course, this model came out of the field of psychology, but it's supported by a lot of direct measurements that neuroscientists have made across many sensory areas. But for example, in visual cortex, if you look at the trial-by-trial variability of individual neural responses to a stimulus, you often find that the trial-to-trial variance is higher than the mean response, so-called super-Poisson variability. So roughly speaking, the signal-to-noise ratio, if you just look at individual units of computation of neurons, the signal-to-noise ratio is around 1. So noise is like an unavoidable feature of biological circuits. So I think already just with that background, there's interesting non-trivial consequences that are worth maybe unpacking here. So over the next couple minutes, I'm going to walk you through building a simple circuit model for how you might perform the task I was showing you before, where you're interested in discriminating between two sensory stimulus. Stimulus A is tilted to the left, and stimulus B is slightly tilted to the right. So the way that you typically build up a circuit model would be to assume that there's a noisy pool of sensory neurons. These could be kind of classic Hubel-Wiesel orientation selective neurons in visual cortex, for example. But again, each one of these neurons has a very SNR around 1. And if I want to think about the simplest circuit model that wires together the sensory pool to an output neuron that only fires in response to stimulus B, but not to stimulus A, the simplest way I can wire them together is to assume the synapses and connections are linear, and then finding the optimal set of synaptic weights ends up mapping onto fitting the optimal linear decoder, linear classifier, right? So this is a toy model, admittedly, but it's one that's quite standard. So this model is actually pretty interesting. It took me kind of an embarrassingly long time to understand some of the subtleties here. So let's even just think about a really simple case where there's only 2 neurons in your sensory pool. So X1 and X2 are the firing rate of these 2 neurons, and then W1 and W2 are the synaptic weights or outputs, and these could be excitatory or inhibitory synapses. And if you think about, here's some example data where neuron 1 fires more in response to stimulus A, and neuron 2 fires more in response to stimulus B. So the 2 neurons are doing opposite things. So if you think about how to optimally wire together the circuit, you might think that, okay, an increase in firing rate in neuron 1 decreases the probability of stimulus B being shown, which is true. And neuron 2, an increase in firing rate increases the probability of stimulus B being shown. However, when you fit the optimal linear classifier to this example, you'll find that both of the optimal synaptic weights are positive, i.e. excitatory. It's not the case that one is excitatory and one is inhibitory. And if anybody has seen Simpson's paradox before, this is more or less a manifestation of a similar concept. So this is the reason for this kind of counterintuitive result lies entirely within the noise. So if you think about the data set in firing rate space, the x-axis here is the firing rate of the first neuron, or the number of spikes fired by neuron 1 on each trial. And the y-axis is the number of spikes elicited in neuron 2. Each point is a different trial, and the color of the points correspond to stimulus A versus stimulus B presentations, and you get two probability distributions. Now, the optimal synaptic weights are what defines this linear decision boundary, which is the black line in this figure. And the point is, by keeping the mean responses the same, but playing around with the trial-to-trial noise, I can move this linear decision boundary, or the optimal linear decision boundary, around quite a lot, and I can change the slope from positive to negative. So this is just to say, if you were only to look at the mean responses of each neuron and ignore the noise entirely, you would completely miss the way to wire the circuit together, at least from an optimal standpoint for this task. Okay, so that's the first part of the talk is already out of the way. The take-home message of this is just to say that even in these really, really simple settings where you have two neurons, two categories that you're trying to distinguish, noise is playing some pretty subtle and important and has non-obvious impacts on what the optimal neural computations are. So hopefully that'll be a hook for the rest of the talk. And hopefully it's also obvious that this is just kind of scratching the surface of what noise is doing in more complicated deep networks, for example, or in biological systems that have recurrent dynamics. So why don't we understand more about noise? And that's what I want to talk about for the rest of my time. In the first part of the talk, I'm going to talk about a real practical challenge for why neuroscientists don't understand more about the structure of noise in neural data. And this is a data analysis problem, which is that we have limited trials, and having limited samples from a neural system makes it difficult to estimate the statistics of noise. So if you think about recording from, for example, 1,000 neurons, this is something that we can do these days with modern recording techniques. If I want to estimate the mean response to a particular stimulus condition, that's like estimating a point in this 1,000-dimensional space. And that's difficult enough, but if I want to estimate something about the spread of the distribution, well, the second moment of the probability distribution is the covariance. You can think of that as an ellipsoid in this 1,000-dimensional space. And to estimate the scale and orientation of that ellipsoid, I need to estimate all of the major and minor axes, which quickly becomes very challenging. If you don't like to think about it geometrically, you can also just kind of count up the number of free parameters you're estimating when estimating an individual vector versus a symmetric covariance matrix in a recording of 1,000 neurons. All pairwise correlations between all neurons ends up being roughly 500,000 free parameters that you need to estimate. And most neuroscience experiments, you're lucky if you have on the scale of thousands, maybe 10,000 trials. So you have many fewer samples than you do free parameters that you're trying to estimate. So this puts us in a challenging statistical regime. Okay, so that's the first problem that we're going to tackle. The second problem that I'm going to come back to is just that even if we could estimate things about noise, I think there's a lot more that needs to be done in order to figure out what are the right things, downstream analyses to do to make sense of it. So if we go back to like simple models where like a deterministic feed-forward neural network, where there's no noise, I put in an input, I just get an output, a vector in this high-dimensional activation space if I look at a hidden layer of this network. So in a sense, the neural representation can be thought of as a point cloud in this representation space, or maybe, if you will, a manifold within this firing rate space. What I'm advocating for is that in real biological systems, and also, by the way, in many modern architectures in artificial intelligence and deep learning, the picture looks more like this, where you put in an input, and then what you get out is a sample from some high-dimensional probability distribution. And working with these kind of probability distributions or fuzzy point clouds is a much more challenging thing to do. The picture on the left, I can do PCA, I can do clustering, I can do a lot of off-the-shelf analysis methods on. The picture on the right is more complicated. Okay, so before I get to that, I want to back up and talk about how can we even estimate noise in neural data. So I'm going to tell you about work that was published at NeurIPS in December of last year. This is work that was led by Amin Najakbash, who's a postdoc in my group, and a graduate student, Isabel Guerin. So we focus on neural data analysis in this particular paper. We look at a few different data sets, but for this talk, I'm just going to highlight one data set, which is recordings from primate motor cortex and premotor cortex. And the animal is doing a very canonical classic task within the field of systems neuroscience. So I'll describe it to you briefly. The monkey puts its hand out and starts each trial by placing its hand in the center of a screen. And then a target comes on, a blue dot, which tells the monkey where it needs to reach. But the monkey doesn't immediately reach. It has to finish out a random delay period, has to wait for the blue dot to turn to a green dot, which is the go cue, and that's when the monkey executes its reach. Okay, so for this task, we're actually going to look at the period right before the motor event or the motor action is executed by the monkey. And we're going to look at this preparatory activity when the monkey's planning its upcoming movement. For those of you who like a sports analogy, I like to think about runners at the start of a starting line just before the gun goes off. So they're in this primed, ready state, and they're just waiting for the go cue. And I should mention that this is experimental data that was collected by Saurabh Vyas with help from Nir Evan Chen many years ago when they were in Krishna Shenoy's lab at Stanford. The nice thing about this particular data set from the Shenoy lab is that they trained a monkey to make reaches to many, many different targets. And because they tried to densely tile 2D space with where the monkey was reaching, they have relatively few trials per condition, which is the statistically challenging regime that we were interested in tackling. Okay, so here's the core idea of our paper. So remember, we're trying to estimate what is the mean and covariance of the neural population response in this delay period activity right before the monkey executes its movement. And the idea is that if the monkey is preparing a reach to location X, well, theoretically, if it was instead preparing a reach towards X plus or minus epsilon, that distribution should be almost the same. And it should be exactly the same in the limit that epsilon goes to 0. So this kind of smoothness assumption is what is going to make everything work. If you think about what's happening in neural firing rate space, the average activity right before the monkey makes the movement, if I change the target a little bit, should not change in firing rate space very rapidly. So if the monkey is making a reach to a ring of targets, the mean response in neural firing rate space should trace out a one-dimensional ring manifold. Okay, and moreover, not only should the mean response be a smooth function of the condition, but the noise, the higher order moments of the probability distribution should also change smoothly. So the covariance ellipse should change smoothly in terms of its orientation and overall scale. And again, I'm showing you this in motor cortex just as a concrete example, but we also look in the paper at visual cortical responses where we smoothly parameterize or smoothly change something about the visual stimulus that the animal is seeing. And in fact, this is a very broadly applicable motif in experimental neuroscience. So if you look in olfactory responses, for example, I can vary the concentration ratio of 2 different constituent odors. And as I do that smoothly, the response to presenting an odor in olfactory cortex will also vary smoothly. So we think that this is a broadly applicable tool that can be applied to many areas of like sensory and motor neuroscience. Just briefly to give you a little summary of the method, the first step of our method is to do a nonlinear regression to predict the mean response. And we use a Gaussian process regression for this. This is a very standard and well-known methodology within machine learning. People have also used it within neuroscience before. The high-level idea is that you put a Bayesian non-parametric prior over smooth nonlinear functions. You can do basically hyperparameter tuning in a variety of ways to figure out the length scale of the smoothness here. And then conditioned on observed data, you get an estimate of what the true mean response would be in this case across conditions. So this is how we estimate a smooth mean response across the neural population. And then to estimate a smooth response at the level of the noise or the covariance, we use what's called a Wishart process. This is also something that exists, although it's a little bit less broadly known within the machine learning community. And the high-level idea of this Wishart process model is that we're going to put a Gaussian process prior on the principal components of this covariance matrix. That's the idea in a nutshell, at least. And actually, what we end up finding works the best is to use a structure like this, where the covariance matrix, the neuron by neuron covariance matrix, has this structure, which is UU transpose, where U is a low rank factor matrix, and plus a diagonal term, which makes the overall covariance full rank. So for the aficionados in the audience, you'll see that this has a feel of factor analysis to it, but we're putting a smoothness prior over the low dimensional factors. That's the basic idea behind the method. I'll just tell you that this works extremely well. So for example, if the monkey is making reaches to targets aligned along a vertical line like this, you see that the mean response and the covariance estimates we get out are nice and smooth. And if you compare this to a regularized estimate of mean and covariance that doesn't leverage smoothness, you get principal component projections that look like this, and we can validate using cross-validated held-out data that our model is doing a better job of estimating both the mean and covariance of the neural responses. Okay, and you can push this really far. So this is one simulated experiment that I think makes the point pretty strongly, where we are looking at we're looking at experimental conditions that are aligned along a ring. And we're only giving the model a single sample per stimulus condition here. So nominally, there's actually no information about trial by trial variability because you only see a single trial per condition. But we're really densely tiling the condition space. And because of this smoothness assumption, the model is able to get an accurate estimate of the covariance matrix by pooling statistical power across nearby conditions. Okay, so your effective sample size per condition is greater than 1 because of the smoothness assumption. Okay, so we're very excited about this, and there's a lot of experimental applications that we're following up on. But in the last 10 minutes of the talk, I want to advertise another bit of work, which is kind of at least gives you a sense of what are the next steps once we do this estimation. So remember I told you that even if we knew properties about the noise, it's challenging to know exactly what to do with them. So in a deterministic neural net, again, I can think of a hidden layer representation of a series of input images as a labeled point cloud or manifold in this high dimensional space. And again, what I'm advocating for is that we broaden our mind a little bit to think about this more complicated scenario where each input gets mapped not to a deterministic point, but a probability distribution over responses. And by the way, a lot of stochastic deep neural networks like this actually are really important and central to many state-of-the-art methods in machine learning. So more recently within the generative AI community, things like variational autoencoders and diffusion models are all kind of predicated on cleverly injecting noise into models like this. But if you go back decades, people were already injecting noise into deep neural networks as a way to regularize them, and empirically seem to improve performance. So I think, again, this is a topic that I thought would be interesting to talk about today because it does seem like there's some intersection here between psychology, neuroscience, and AI on this topic. Okay, so I'm not going to completely solve this problem. I think it's an open problem in many ways, but I'm going to narrow it down to a specific question, which is, if I were to measure the mean and something about the noise statistics of across multiple neural networks. So for example, I could record from 3 animals performing the same task in neuroscience, or I could train 3 deep networks from different random seeds or with different hyperparameters in a machine learning setting. How would I know whether the networks are learning similar representations or different representations? And this is a very well-studied problem in neuroscience as well as in deep learning. in deep learning, it's been very well studied for the deterministic setting. So I'm going to take a step back and talk about that. And when I say measure how similar different representations are across neural systems, I mean that in a pretty literal sense. I would say this might be a slightly controversial slide, I know, for some people in the audience, but for work that's been done in my group, I've been advocating for certain analyses of neural networks that require you to think about distances in a pretty literal way. So I'm going to think about measuring the distance between network A and network B using distance measures that give you a value of 0 if and only if network A and network B are equivalent by some pre-specified criterion. I also want a notion of distance that is symmetric. So the distance from network A to network B is the same as the distance from B to A. And then also, I want to satisfy the triangle inequality. So the distance from network A to network C is always less than or equal to the distance from A to B plus the distance of B to C. Now, I'm not dogmatic in saying that we always need to satisfy these things, but for certain kinds of analyses, like if I want to do clustering across networks or I want to do nearest neighbor regression, having these properties is pretty useful. And from my perspective, I think there's a lot of ways that we can get these properties without sacrificing too much in many cases. So in, again, the deterministic neural network setting, one way to get all of these nice properties is to use what's called the Procrustes distance. So the idea here, again, is we're thinking about two networks as being colored point clouds in this high-dimensional space. And the problem is I can't directly compare these point clouds because the neurons in one network are not going to be labeled the same as the neurons in the other network. So at the very least, there should be a permutation nuisance transformation that I want to remove out. The Procrustes distance goes a little bit further and says, instead of removing permutations, let me remove rotations. So I'm going to find the best possible rotation matrix that aligns these two matrices, these two point clouds, and then measure the distance between them. So you can think of this as a linear regression, but with an added constraint of orthogonality on the regression coefficients. This is just one more view of that, but you can generalize this, and this is something that we've pointed out in past work. You can generalize this in various ways. For example, if you pre-process the data by whitening the point clouds and then apply a rotational alignment, this also gives you a metric, and it ends up being closely related to canonical correlations analysis. So these two different metrics, you would use them if you wanted to be invariant to different sets of nuisance transformations, like rotations versus any linear transform or affine transform. OK, great. That should be fine. So very quickly, what I wanted to tell you about today is we were stuck a while on how could we generalize this to the stochastic setting. So now we don't have point clouds that we're comparing, but we're trying to compare two fuzzy point clouds, if you will, or probability distributions that are aligned to inputs. So in this paper that was co-led by Linden Duong and Jingyang Zhao, we found a way to generalize the Procrustes distance by effectively fitting the optimal rotation to align these probability distributions across networks. And then the question is, what do you use instead of Euclidean distance, which you can no longer use between probability distributions? But there's two nice generalizations of Euclidean distance that turn out to work. One is the so-called Wasserstein distance, which is an optimal transport distance. You can think of this as the amount of energy it takes to move one distribution onto the other. And then the other is called the energy distance. This is a nice distance function between probability distributions that doesn't suffer a curse of dimensionality. What's nice about both of these ways of measuring distance is that in the limit that the variance of the noise goes to 0, you recover the Procrustes distance. So these are bona fide generalizations of the Procrustes distance, which is a reasonable metric in the non-stochastic setting. Maybe I want to leave time for questions, so I'll breeze through this application, but we're applying this to deep neural network autoencoders, which are becoming popular within neuroscience and many other fields of biology to be a general purpose dimensionality reduction tool. And so the basic question is, if you train two networks, I don't know why that's happening. Okay. if you train multiple networks on the same data set, do they find similar low-dimensional representations, or do you get different answers when you train different models? And one of the nice things about these distance metrics is that they scale to very large data sets. So here we're looking across 1,800 deep neural network models, and we can fill out this distance matrix, which tells you how far every pair of networks are from each other using this generalized notion of Procrustes distance. And this is, again, symmetric, and all these distances are self-consistent with each other in some sense due to the triangle inequality. And you can do things like try to find low-dimensional projections now, where each point here is a different deep neural network. And you can color them by different points and see that they cluster. You can also do supervised learning analyses, like predict things about the network architecture from where it sits within this space of networks. So I'll go past that. I won't belabor it, and just give you a couple take-home messages so we have time for questions. I started off this talk by telling you that I think noise is really interesting, and I don't want to say it's understudied, but I think it's understood because it's a very difficult thing to study. Even in really simple cases, it has these counterintuitive effects. One of the reasons we don't understand that much about it is because just estimating it, at least in neuroscience experiments, is really difficult. I showed you that we can exploit Bayesian non-parametric models to predict noise distributions with limited trials. And then I also introduced some new tools that I think can help us do kind of large-scale analysis and comparisons of the stochastic structures of many deep network models. And with that, I'll take some questions. Yeah, questions?
So I'm curious about your perspective on what counts as noise.
Yeah.
So
it's a loaded term. I'm just using it in the sense of any sort of stochasticity, and maybe I'll let you finish.
Measuring, at least what you showed in your slides, is variance across trials.
Yes. Yep.
Right?
Yep.
And that's not necessarily stochastic. I mean, stochastic from the standpoint of the experimenter, but it might be stochastic from the standpoint of the brain, right?
Yeah.
So do you have a perspective on how much of that is actually stochastic from the standpoint of the brain, and how much of that is the mental state? Or
no, I'm trying to think about it from this really general perspective without, to answer that question would require talking about specific experimental setups and stuff. I guess I think what you're getting at is that in real neuroscience experiments, there may be things that look like noise, but really it's a confounding variable that you didn't measure. So the question, yeah, I think that's a good comment, and it's still interesting, I think, to study noise because there's always going to be quote unquote confounding variables, and probably confounding variables that the brain doesn't have access to in a sense. And also just in purely artificial systems, there is noise injected. So I think it's a good point that some of the noise is there for bad reasons, but some of it is irreducible, I think, in those cases.
Yeah, so I actually have a very similar question, but maybe not so similar, actually. This might be quite silly. So, how do you know that the noise, the so-called noise, I guess, that's being measured here is actually something, bad in the entire state of affairs, right? Mm-hmm. Meaning, for example, are you talking about randomness in the brain, or are you saying that there's literally information destroying noise that's bad and should not be there, right? That's so.
So I think there.
I know randomness here is not a feature.
Yes, no, I would not say that. Yeah, I wouldn't make that claim, and I think there are systems like I think one of the famous ones in neuroscience would be the songbird system in zebra finches. Is an area called LMAN that directly, or at least the working model is that it is injecting noise into the bird's behavior so that it can do some exploration and learning. So there's definitely cases where noise is a feature and not a bug. I just happen to motivate it from psychophysics, where people tend to think about noise as information limiting. And I think that's also, maybe in those cases, it probably is more of a bug. So it's very context dependent, I guess.
I have a question about this smoothness. Really interesting talk. This sort of smoothness, if I understood correctly, in the primary reaching study, the smoothness assumption helps to fit the data a lot better.
Yes.
But it sort of seemed when you raised the initial, I was really curious because I was like, it didn't seem obvious in any way that a neural system should satisfy the sort of smooth variation. And sort of think like, I don't know, bring to mind things like sensing or in nerve sort of things. We sort of found like injecting really high frequency, like fast oscillations is sort of works better for that particular neural network. So any more comments about the what it means?
It kind of goes back to this psychophysics idea that if I were to move the target that the monkey was reaching to epsilon, perceptually it wouldn't be able to tell the difference between x and x plus epsilon. So I think that's the kind of justification for some smoothness that.
But couldn't the neural network that's processing that change have very high frequency variation?
It's possible, yeah. It's possible that the neural systems could just have super perceptual fidelity for these things. And we can set, using cross-validation, for example, we can learn the optimal length scale and try to ask, like, what length scale is the neural system operating at, and does that match with psychophysics? You might be able to actually align the two.
Maybe one last question, and then we'll have our break.
Yes.
I was really intrigued by the plot with the neuron and the signal to noise ratio about 1. It's one thing that's maybe long enough on Bruno, like a biological system trying to compute the most efficient way possible. Might figure out how to, in some sort of channel coding way, figure out under some power constraints the optimal signal to noise ratio.
Yeah, I actually meant to say that in response to an earlier question about a feature or a bug. Like, potentially one feature of this is that, it's more energy efficient and it doesn't hurt your perceptual performance enough for it to matter. So yeah, does that answer your question, or just kind
more
of?
to comment on that?
Yeah, I mean, I would not consider myself an expert on these kind of energy efficiency arguments, but I think that definitely there would be people who would advance that perspective. So I think it's a good comment.
Great. Let's have the break now. So let's thank Alex again. And we'll come back here at 3:35 for Phil Freeman. So back here at 3:35.